Excellent.
Welcome, everyone.
Okay, well, let's get into this now and kick right off. So obviously, folks, we are extraordinarily excited as we go day by day, getting closer to our first in-human opportunity to show whether and how well our therapy works in humans. We have every confidence that it will be. We've done a ton of studies, as you've seen. We've been releasing different types of cancer models. Everything looks extremely positive. We have, as promised, delivered a mountain of data on the safety side, more of which was released in a press release from the company this morning, indicating that there are no endotoxins, again, in our batch of materials that will be used in the human study. We are pending right now approval from an ethics committee. Once that is received, we will be in a position to start enrolling, initiate the study, and then the big announcement for us would be the first in-human dosing. The first dosing would be a critical announcement for us leading forward. As we go forward, I have to say, in both Carmen's lab and Sona's lab, the excitement is palpable. This has been something we've been working on a long time. Investors have been patient. But we're beyond excited to move into it. So today we want to go through a few items and a few updates we have for you, and look forward to doing more of these and having more news for you in the future. So first of all, of course, forward-looking statements. We're going to make a number of forward-looking statements, so we're going to refer you to our disclosure documents and the risks and unknown risks that are identified in them, of course. So today, very brief update. Number one, overview on our THT therapy. Many of you heard it before. We're going to do that. There are prior webinars that go in depth into the history of targeted hyperthermia therapy, how it works, the mechanisms of action, etc., how we got here, and of course, all the tremendously promising preclinical data we have. What's new for today is we've got some endotoxin study results that were just announced in a press release we'll talk about. We recently released a research agreement we've struck with another company on an interesting technology that may help to make our targeted hyperthermia therapy work better and faster. And we have some images of our next generation laser that has been completed and is now ready to be deployed in our human study. Finally, where to next? That's what everyone is interested in. There's an ASCO presentation, a major cancer conference, and of course, the first in human early feasibility study. We'll have a little bit more information on that, and then even looking further down the road on the future IP that's being developed every day in our lab. So Sona makes gold nanorods. Many people make gold nanorods. We are the only ones in the world who make them. We have a patent for making them without the use of a toxin. That's important when you're going to put trillions of these into a body, which of course is what our therapy is based on. Gold nanorods themselves are optimal amongst nanoparticles because of their ability to convert non-thermal energy into heat. We do that with a near-infrared laser, and they're just very efficient. What does efficient mean? Why is that important? When you bring it down to the clinic, it means that when we're treating cancer patients in clinics, it can be done in a short period of time. The shorter, obviously, the better from a clinical and an economic model perspective. As we say, ours are uniquely biocompatible in that they're the only ones made without a particular toxin. That makes them perfectly suited for nanomedical applications in the body. And we've talked much in the past about the fact that these parameters have been validated by the nanotechnology characterization laboratory that was set up by the FDA and the National Cancer Institute in the US to assist companies like ours in nanomedical areas to further their research and their products. Apoptotic cell death, you see, this is what we're after here. I'm going to let Carmen talk a little bit more about the actual mechanisms of action in our therapy, but suffice to say that certainly the foundation of the company is what we see as a platform technology in these gold nanorods because, well, our first application is this cancer therapy. We are working on and have some progress, at least in a number of different applications for it, making it, as I say, a platform from which to grow. Carmen, do you want to talk about what the problem is we're seeking to solve here?
Yeah, so the world is well aware now that immunotherapy in cancer is essentially the mainstay for so many of the cancers, not all of them for sure, but so many. And it's largely because we know that the body actually sees cancer as foreign, as a threat, and innately attacks it. The problem is, of course, is that the cancer also has conflicted messages and the body is attacking itself. And so there's reasons for it not to pursue the immune attack. And then there's other reasons why it fails, and that is that the cancer in and of itself has developed ways to evade immune detection, so evade that threat. So pembrolizumab, one of the world's leading anti-cancer immunotherapies, it's a PD-1 inhibitor, a programmed cell death inhibitor. So suffice it to say, it's the primary focus of all immunotherapy today, and in its best response rate, 40% of patients respond, and the durable responses are somewhere between 21 and 30. And so this, that's fantastic. Many of these people had nothing prior to this, particularly in the world of melanoma and other cancers like that, where literally no chemotherapy ever worked. That's exciting, but that means that there are somewhere between 70 and 80% of people that still need a better solution. And so if you look across the board, kidney cancer and melanoma are big responders. GAT has some, but that's a nuanced statement. But then when you move to the right past lung, that NSCLC, it's the non-small cell lung cancer, then these numbers drop dramatically. And of course, we're seeing little to nothing in things like colorectal, pancreatic, hepatobiliary. And so these cancers, if you look at colorectal cancer, breast cancer, these are and lung cancer, these are the top 3 killers in the world, and we're still struggling at 20% or less, and far less in colorectal cancer. And so these are challenges, but we're all aware, those of us in the immune therapy world are aware that these challenges are surmountable. And so this is the goal of the targeted hyperthermia.
I'm just going to check that there's a volume check here because we had one comment. And by the way, there's a great opportunity to point out that there's a chat button here in Zoom that you can put any questions into, and we'll respond along the way as we go. Yep. There was a question here, was there volume? Want to make sure folks are hearing that. And there's some more questions about our pathways through regulatory paths as well.
I apologize if my volume's down, and I'm not getting an indication on my side, but no. Please do let me know. I've never been accused of talking too quietly.
There we are. Okay, well, let's circle back towards the end on regulatory pathways. We've got a little bit of a discussion around that and where we think we go from here. But flicking right along here. You want to take us through this, Carl?
So yeah, how does this work? And we've gone over this. Some of you, probably many of you now, are aware of the steps, but essentially we use preclinical models to prove the immunotherapy. By the way, preclinical models are the foundation of all immunotherapy. The first PD-1 inhibitor the Nobel Prize was awarded for was in a mouse model. So what we do essentially is we take mice, because mice have immune systems that mimic ours in many ways, and we can learn plenty of things about how they respond. We grow tumors in mice, and we take the most that will talk. These tumors, the cells, the cancers are actually from these mice. These are cancers that are generated by these mice, and then we take healthy mice and reintroduce the cancer to them. We inject them typically in the flank, in the subcutaneous, in the skin, just the area just within and below the skin, to create a tumor. And when the tumor is palpable, when we can feel it, then we can target it. And so what we do next is we take our gold nanorods that David explained. The gold nanorods are 60 by 15 nanometers. You know, a hair is 80,000 nanometers wide. So these are in minuscule little things. And we inject it, we infiltrate the tumor, wait for it to kind of settle in the tumor. And then we pass a near infrared light, a laser, over the tumor for 5 to 7 minutes. And the near infrared light passes through the nanorod, and in doing so creates energy within the rod that vibrates, and that like a tuning fork if you tap it on a desk. And so that vibrational energy creates friction, and friction creates heat. Heat is the essence of it's the main ingredient in this whole story. Heat is, we've naturally evolved so that when the body is threatened by things like infection and even like some tumors, we generate heat. Now, our system generates heat. It's not effective enough to kill tumors generally, or to cause a cell death, because the heat we need to create is like a high fever. So we target the heat to 42 to 48 degrees. That is high fever heat. And in that range, tumors, or our normal tissue will tolerate that quite nicely. But tumors are stressed by it. And in fact, there's a whole pile of biology we're not going to go into today, but I welcome anybody to, call me up and talk about it. But we take advantage of this heat shock protein pathway. That heat shock proteins are liberated when we pass the heat through the tumor, and the cell goes into a thing called an apoptotic cell death, which means it dismantles itself. We know that every day cells die by a programmed cell death. We replace our cells. Every one of us that are on this call is not the same person that was born, for the most part. All of our cells are new. So there's a renewal process. What happens with this is the cancer cell is triggered into going into that death cycle earlier. And when a cell goes into a death cycle, it's not like taking a hot poker and brazing the thing. That's not ablative. What a cell does is dismantles itself, and all the building parts are repurposed and presented to the immune system. And so in the process of presenting those dead cell parts to the immune system, if the dead cell parts are broken, like a cancer cell, then the immune system will recognize it. And so what we're doing with the THT is literally showing all the broken parts to the immune system, and the ones that are particularly abnormal are the ones the immune system will target. And that's what we've proven over and over and over again through this process. We can exploit that natural process. So this is targeted hyperthermia, and it's targeted meaning that we are literally just heating the tumor and its microenvironment, the center of it. We're not heating the whole body. So as a toxicity, this is extremely well tolerated by patients. We do in historically and still in operating rooms that I work in, we use hyperthermia, but we heat a whole body, which is extremely stressful to the patient and takes a lot of. So we've eliminated all of that toxicity by targeting the tumor directly.
And in past webinars, we've shared a great deal of our data on the efficacy studies that have been conducted in multiple cancers. And that data just continues to build. We are continuing to do studies every day. And Carmen, the big question people have is, what is the probability that this can translate from these incredibly promising mouse studies into humans?
Yeah, so this is going to sound like bragging. The probability is 100%. Now, why am I saying that? That's overreaching, and I'm a zealot. But here's the deal: I have treated every tumor we have treated with this, and I've treated humans as well. Remember, I treat people with hyperthermia. It's just a crude way of doing it by heating a whole leg, or heating a whole arm, or heating a whole belly. But this works. So we've treated breast cancer, melanoma, colorectal cancer, 2 kinds of colorectal cancer. Every tumor 100% of the time responds. Are we going to see, yeah, it is going to work. At the extent to which I don't, over-promising and under-delivering is a bad idea, but 100% that we will see response rate. If we see a response rate that's over 50%, we should be ecstatic. I would venture to say that.
And what's the difference between achieving a response rate and actually curing someone of cancer? That's, I think, an important distinction here, right?
Well, that's the essence of it is this. It's a complicated question, in fact, but if we see a response rate, it doesn't mean the response will follow through to clearing the cancer. But you looked at the pembrolizumab map data. Yeah, they have a response rate of 40%. They have a clearance rate of half of that. And so the response, you can activate the immune system, but you have to activate it in a vigorous way. And I don't want to use jargon, but we need a strong message. We need a strong antigen. And so the targeted hyperthermia, the hyperthermia presents all of the antigen to the immune system all at once. And so what we've learned through our research is when we do that, at the time that we've exposed all those antigens, if we now even further enable the immune system, we get a massive response. And so the response rate is indicative of the likelihood that we will achieve cure. The higher the 100% response rate means we will achieve cure in something less than 100% for sure, but hopefully in 60 or something percent.
And I think what's most relevant is the fact that you cite those Pembro or Keytruda figures that are cited, and that there's a 60% response rate, but a 20% cure rate, clearance rate, I think you called it.
Right.
Everything we've seen in our preclinical studies is we're having higher response rates than that. Therefore, our expectation is that we'll have higher clearance rates too.
Exactly.
So it's a step forward from current practice. That's why we're so excited, and that's
exactly right.
Good discussion. So moving forward, what's new? Number one isn't listed on here, but we should mention, we're honored that we have our first equity analyst research pickup from Maxim Group. Now, that's a 20-page report that's been written by a top-notch team led by Jason McCarthy. There is a PhD, has a very strong understanding of the science here. And while that's, we have to legally not endorse that per se, that is their study, it is their conclusions, their data, etc. We are still honored to have them. And you may have noticed on our website, we now have a page for analyst coverage, and they're the first to pick up. And that we think is going to be very helpful for a number of reasons for the company moving forward. But their conclusions and their projections are theirs alone, and not the company's per se. Nonetheless, it's a great thing to have for the company. First moment to have first analyst coverage. Now, jumping into the endotoxin study results that we mentioned today. This endotoxin is something that we've talked about in the past because the the Nanotechnology Characterization Lab, some longtime followers will have seen, have done analysis on our materials before and found them to be absent of microbial matters and endotoxins, importantly. We've now gone to a second source for this, specifically for this batch that is going to be used in our clinical trial. I don't know, Carmen, do you want to speak any more as to the importance and what endotoxin is briefly and what?
Well, yeah, so very briefly, an endotoxin is a contaminant. An endotoxin is a part of the bacteria or a bug that causes us to be sick. When we get an E. coli infection, it's E. coli endotoxin making us sick. We have to be assured that we're creating these gold nanorods in the cleanest, purest way, and those samples have to be evaluated to make sure that they haven't been contaminated. And you can imagine it doesn't take much to contaminate these batches. So there's a lot of rigor that goes into the preparation and the packaging, and we're happy to see that so passing the endotoxin assessment enables us to one step further to clinical practice.
And every time we emphasize that our materials are biocompatible, this is what we're relying on. We're not relying on ourselves, our own assessments, our own technologies. We go to third-party labs. We said we'd build a mountain of data, and that's exactly what we've done. And this is another study that will be used in our regulatory submissions, all of which give us great confidence as their ability to be accepted. We have mentioned in the past that there is also a part of this mountain, a Toxology study. This is still pending. And it's we talked about it last time as critical prior to our first in-human study. It is pending. We're not concerned about the timing on it. We're not concerned about the results. We're excited for them, in fact, and we'll look to report those as soon as we've got a final report from them on it. Carmen, you want to speak to Toxology?
No, I think, our early indications, and, again, I don't want to be crystal balling this stuff, but at the end of the day, our toxicity indications were that they were extremely low. And again, we've taken steps through this method of direct injection and targeting tumors specifically. We've limited the exposure to the host. To the point where we're quite confident these are going to be good. And we're excited to see these results coming in to confirm that what we believe is right.
That's it. Just another one. So next, BioVaxis research agreement was a press release from last week or so. Perhaps, Carmen, you can take us through what this is all about.
So the BioVaxis has a technology. It's essentially a lipid-based. So a lipid is a fat molecule-based delivery system. Lipids, to really cut to the chase, lipids are one of the things that will stimulate the immune system in a non-specific, crazy way. So it heightens the immune response like a booster. And so essentially it was designed for encapsulating these bioactive molecules. What's a bioactive molecule? If a cancer has a quirk on it, if it has a broken protein, if you could take that broken protein and amplify it and put it in this kind of lipid emulsion, it would increase the chances that the immune system sees it. So it's a very attractive technology for us, because in fact that's exactly what we do. We expose, and this is unique to us for sure. Nobody's doing this. We are liberating antigen that has not been liberated. That's why all of our research shows that our THT works in situations where standard immunotherapies haven't worked. They just haven't seen antigen. We show antigen, it works. So we're looking for ways to optimize that. And we have some reason, we actually have people working in my lab that worked on the development of this DPX. And so we have some fairly good insight into the biology of this, and believe that we can benefit, we can enable this even better than it was initially meant to be.
Yeah, and this is a quick research agreement. It's not a development agreement. This is, let's see if there's some evidence of a synergy here that can really add something to what we have, which is already powerful. And if that works out, great, we'll figure something out on that basis. And if not, fine, we'll move on. It's important for us to be pushing forward and pushing the advantage we have and looking for these opportunities, which we will continue to do with the scientific team and other possible partnerships. So this is our brand new next generation laser for a device, which is all set and ready to ship for our clinical trial. A lot of work has gone into this. A lot of the last webinar, we talked about some of the validation and verification work that's required to do of it to effectively establish and be able to share with ethics committees and various other regulators that what we say it does, it actually does. That work was done by a top-notch company in Canada. And we're very, very pleased with that validation, the results and the end unit. And we've made a number of enhancements to it. That's the hand piece on the side. That fiber you can see on the right is literally the fiber the laser passes through. And this makes it an easier hand grip for the operator to use. Carmen, you've played with this a little bit. Do you want to make any comments on the new version?
Yeah, this is a very elegant device. It's nimble. It's very reliable. We've confirmed, the company has confirmed that the energy is reliable. It's consistent. It's got all kinds of built-in safety measures, and so we can adjust the wavelength that we use is 860 nanometers, and so at that wavelength, that's an optimal wavelength for treating tissue. It penetrates 2.5 centimeters or so, and my American friends reminded me late yesterday, that's an inch. And so it penetrates an inch into tissue, and there is, if you look at any cavity in the body, we can either target a tumor from the surface or from some lumen. like a colon lining or or urethra and a prostate or a rectum. So this gives us lots of access to tumors, and without the invasive component that every other technology needs to get there. And so that's another. So this machine is super well crafted. The simplicity of that handle, if you've ever been in a clinic, to be able to hold a unit like a pen over a tumor is both relaxing and comfortable for the treater, and gives it lots of flexibility as to where you can aim it on a patient. And we actually have a kind of a little tripod fancy stand that holds that up. But the long and the short of it is this is a very comfortable device for a treater. And when we make these kind of things, you need utility and consistency, and this we're very excited about this. So that's another Sona development. We haven't talked about it very much because it's really the nuts and bolts of it, but at the end of the day, this is an improvement. And that wavelength is, if any of you have looked into laser technology, you'll see this is the optimal wavelength to treat these things, to use for this purpose.
And that's why we needed a custom made laser. This was not an off-the-shelf possibility, but if you want to get that depth of penetration, this is what was necessary. And so very pleased with what we have. And as I say, as Carmen says, it then allows us to go into any number of different types of tumors in different locations. Of course, starting with melanoma in this first early feasibility study with humans. But from there, we already have plans to move on to others. I'm going to just pause here because there are a number of questions. I want to try and address some of them. Carmen, do you believe possible outcome with BioVaxis research could lead to a personalized cancer vaccine of sorts in time?
So great question. With or without BioVaxis, the technology that we have can lead to a personalized vaccine. Right. So the important thing is, again, when we're generating these neoantigens, when we're dismantling a cancer cell and showing all the protein to the immune system, we are essentially personalizing the vaccine. I'm going to show you some data in a few weeks that prove that concept in the animal experiments we're doing now, the preclinical models. What we hope is that the DPX will increase the incidence of that experience. So it can, if we can optimize that even 10% further, that makes us even better. But we have, again, I'll show you the data, but the answer is with or without the DPX, we will in the not too distant future be talking about personalized clearance. Curing cancer is essentially, with an immune therapy, is a form of vaccination.
That's it. And of course, this is our, we refer to this as our laser, but the reality is this is a near infrared light that we're using, uses laser technology, but it's not the same as the type of laser you'd have in a surgery doing cutting. Pointing this, the skin does not cut. This is just done with near infrared light in order to pass that gentle non-thermal energy through tissue that then hits the gold nanorods that have been injected in the tumor. They start to vibrate and cause heating the tumor from the inside out again.
And that's an important point. I'm just going to jump in. That this makes it speaks to the safety of this and the wide the wide applicability of it, because at 850, 860 nanometers, you're getting to the top point before you have to start worrying about superficial tissue damage. All of the lasers in the ORs that we use to cut and ablate tissue are literally cutting and ablating. But it is in the laser spectrum, so that's why it's loosely spoken to. But it's the near infrared light end of it.
Right. Great point. Thanks for that point raised. Okay, so where are we going from here? Next stop, frankly, is at ASCO 2025. Carmen, what is ASCO? And here we have our expensive, recently hired models here who will be presenting in Chicago.
Right. Yeah. So ASCO, American Society of Clinical Oncology, it is the world platform for clinical trials, for updates, and for breakthrough. And so the ASCO, the meeting that happens every year in Chicago at this time, is the meeting where breakthrough information and new data is being presented. We're very fortunate to have a poster to present at ASCO this year. We're excited that and I would invite you all to, if you just go on Google and look at our most recent publication. This publication has It's in
Frontiers in Immunology.
Frontiers in Immunology. Thank you, David. Just a senile moment. I apologize to everybody. Frontiers in Immunology. So our article now has had 6,000 hits, over 6,000, I think, 880 downloads, and 2 citations published in January. This is 4 months out. We are now in the top 5% of all journal articles being reviewed by that, published by that group. So we're very excited, and that's of course what got us. We submitted to ASCO before we had those stats. So, and these reviewers, again, this is me being excited and patting ourselves on the back, but these reviewers, when we get an opportunity to present at ASCO, you have something to say. So very excited. And this, of course, as you can imagine, is the optimal networking opportunity for us.
Yeah, everyone is there, as you can well imagine, like 6,000 participants.
That guy on your right to my left is Barry Kennedy, by the way. I just want to introduce him. He's my chief scientist. He is the quality assurance, the superb guy. He translates everything we talk about and develop. And I just wanted to give he's worth a shout out.
Yeah, absolutely, he is. Okay, so what everyone really wants to know about, present company included here, is first in human early feasibility study update. Where are we with that? Where is it? When is it going to happen? What are we doing there? This slide is a repeat from the last webinar. I'll warn you of that in advance. But suffice to say, we are with Ethics Committee now and awaiting comments from them back. That is the necessary critical step required before we can then kick off with enrollment and as I talked about earlier, first dosing. Still aiming to do that early summer. June is the objective here. Still think we're tracking towards that. And so that's why we're just increasingly excited as here we are around May 15th and see that this is in our future, in the very, very near future. Carmen, do you want to speak to sort of our protocol here and what we're achieving, where we're seeking to achieve?
So again, for those that probably haven't heard about this, so an early feasibility study is necessary, as this has never been done in humans. The first step in the study is to identify patients that, now we've selected patients that have been on immunotherapy and have failed. That's because we are, of course, our hypothesis is that we'll prove that we can convert them to responders, the cold tumors or the hot tumor. We need 10 patients and not more than 10 patients. Why just so few? Because really the feasibility study is to demonstrate that we can do this safely, effectively, and efficiently to patients. So we need to know that this isn't an onerous, painful, unacceptably toxic therapy that would shut it down. And so if we can, it takes just a few patients. And so we're very excited about the organization that's doing the research for us. Dr. Rojas is the principal investigator. He presented at ASCO last year. He's presenting again this year. These are world-class medical investigators. And so we select 10 patients, and then essentially what we do is we biopsy part of the agreement and the consent. The patient agrees to have a biopsy done of the tumors that aren't responding, that are persisting, and then we start our therapy applying the targeted hyperthermia. So as you've seen, and I've explained in the mice, injecting the gold, passing the near infrared light through them, demonstrating that we can do this, the patient tolerated it, that the treater can do it, they can learn the technique with relative ease, and then simply observing what the response is to the tumor. Was it painful? Did they mind it? How did they do in the days following? What their impression of it was. And then we repeat the event. We re-inject and we retreat, and on day 8, that's the 2 therapies. Our research has shown us that 2 therapies is optimal, and then we follow them. And so we every week they're followed. We monitor the response in the tumors. We monitor the response in the patient. Are they having complications of any kind? And then at the end of it, at the third, like after 4 weeks, what we want to do, if there's tumor remaining, we'll biopsy it to see how effective was our therapy. What was the immunology that we generated, that we introduced in this manner? It's a short study because that's what's required. While we're talking, we're planning the follow-up study, of course, but we need to get this one behind us so that we can move to the more pivotal studies.
Yeah. And so in terms of what we'll be able to report on, Carmen's talked about the idea that we'd at day 30 be able to assess the tumor. And it's going to be tumor size, but it's also going to be biomarker data taken from the biopsy to be able to understand, okay, great, the tumor's shrunk, but what's going on in the immune system? Have we created the change that we're looking to change here? Because that is the whole concept. Yeah.
And just to frame that expectation, we don't expect tumors to disappear in 30 days. That would be, if that happens, sound the bells, it's a miracle. But we do, there will be some incremental reduction in the size of the tumor, and that's response. And so that if the tumor wasn't responding to everything else that's been going on, that's a win. But then the next win, as you just said, is so then why did that response happen? And then we intend, and our hypothesis is we are showing the response because we've generated novel immunity that wasn't there based on the first biopsy and the comparison to the next biopsy. So
yeah, so I hope everyone gets a sense as to why we're so excited about this right now. It's so close, and it's a great deal of work has gone into setting this up, establishing this, a lot of time as well, as our patient investors will tell you. And we're so close, we expect to be able to have results quickly with a 30-day cycle time for each patient. There's a question here about enrollment time and whether we think it'll be quick enrollment or not. Carmen, you've had this discussion with Dr. Rojas.
Yeah, so Dr. Rojas, they have their patient population is similar in terms of the treatment of the diseases, but they have a large volume. And Dr. Rojas is quite confident that we'll have somewhere between 3 and 6 patients in the first month. Now, I'm going to be that's very optimistic. This person is the expert in that field. If I would be excited if we got 2 per month, to be honest. But his expectation. You see, the thing is with this group, and this is partly by design, many people that are treated with immunotherapy are treated for a long period of time, even if there is no response. So some of the patients are going to be patients where the tumors have stalled. They're not growing, they're not shrinking. And so this is a cold tumor, but it's a stalled response. And so that's an optimal patient to see if we can reinvigorate that process. And then the other people, of course, are going to be people who are growing through their treatment. And this phenomenon of melanoma spreading to the skin and the soft tissue on the surface of your body is quite frequent. And so they have a patient population that actually doesn't have an alternative to what we're bringing to their treatment menu. And so this is why he's quite confident that our numbers will be. We should if we see 6 in a month, that would be wonderful. If we see 3 in a month, I think we've achieved more than we could hope for.
Yeah, the fact that they're on the best option available currently and it's not working leads us to be very confident in terms of our ability to enroll patients into this study.
Yeah.
Okay, great. Where are we going? IP development. There's a pipeline being built out here. We've talked about it in the past. We have a strong set of patents now, provisional filings, continuing to build it out. And there's a bit of a pipeline here we'd like to speak to. Leading the charge is our solid tumor therapy for THT on melanoma. And as you see here, we're through our preclinical studies and just on the cusp here of this early feasibility study. The next stage after that would be a pilot study. The next stage after that would be the pivotal study. This pilot study, we already have the protocol written for it. We're already in discussions with regulators and ethics boards for it. And the goal is that as soon as we are through our early feasibility study, we're ready to go into the pilot study. That will be with melanoma, but we'll be following quickly in suit with colorectal and breast cancer. That's the intention. We also see strong possibilities in glioblastoma, bladder cancer, and cervical cancer. Now, it's not just THT on its own as this tumor therapy. We have a number of other concepts, let's call them here, a THT triconjugate and a theragnostic concept that we can't talk a lot about right now because patents haven't been filed in yet on that. We're working on it still. But suffice to say, we're really excited about these possibilities too. These are all facilitated by the fact that Sona has uniquely biocompatible gold nanorods. Carmen, do you want to speak to some of these?
Yeah, so I mean, this is really the kind of when you look at it as a platform, what are the other ways you can use this? And so these conjugates that we have filed some IP on for sure. This is, when we're talking about liberating antigen, liberating protein from the tumor through moderate heat, having priming the tumor microenvironment to be even more responsive is the goal. And so this is what the conjugates are. It's delivering all of the components. There's in many treatments, in many chemotherapy regimes, nobody gets one kind of chemotherapy. You get a cocktail of chemotherapies because you're looking at the challenge from different perspectives. And so this is what the conjugate research is about, is how many perspectives can we attack at one time in the context of our targeted hyperthermia. That's critical, doing it in the context of the delivery of the hyperthermia. And the rest of it, I think at this point, it's really still on the drawing board. And so we'll wait to describe some more.
All right. And this is a good opportunity to field. There's a question here about the regulatory pathway that we'll be pursuing, because of course we're at the cusp, as I say, of our early feasibility study. To get to pilot study, this would be a North American study in Canada, frankly, that we'd be starting with, and for which we'll need Health Canada approval for that.
Right.
I would anticipate that a pivotal study with hundreds of patients would ideally be done in the US, frankly. So FDA, who we've been in conversations with as well, needs to be on side. Now, using FDA speak, question is specifically, would it be a PMA or de novo path? To answer that question, and there are a number of pathways that the regulators provide, but to answer that, we really need to see the results of the early feasibility study and the results of our toxicology studies. Those two will help to inform whether we can, as we're planning to do, apply for breakthrough status. Breakthrough device status would be the optimal path for us, and that is what we're pushing for, and that's what we think is feasible. If not, the fallback from there is likely PMA. But what is going to matter is what the results are coming out of this study, and how many patients we get a response. And there are several questions also asking about, when are we going to report back on these patients? Is it going to be patient by patient? No, we won't want to do that because it's too anecdotal, one patient at a time. But I also don't think we'll be waiting until we have 10 patients. So we'll once we have a first cohort, let's say, of patients done, and we'll see how many are queued up in the first month that we can get through, we'd probably come back and report that back to folks. These studies, many of these studies, the most successful these studies get shut down, in fact, if you have very effective early results, because it's so clear. We'll see if we're that fortunate. We're certainly optimistic. We've got all the preclinical data supports that it will do, but we shall see. So moving forward in terms of timeline, this is still tracking right now. Biocompatibility feasibility study results, early feasibility results, verified second laser device. We've shown you that's completed. And just going into, hopefully soon, our early feasibility study, and then having readouts in Q3 from that study, and moving on into pilot study dosing, late this year, early into next year, most likely, with readouts next year for the pilot study. That would be the 30 to 40 patient study, which would need approval from a regulator like Health Canada. The tri-conjugate development, importantly, in order to get those patents issued, you have to show enablement. It's not enough to present an idea. You have to show that you can back it up and make it happen. That's some of the work that's going on in the background in the lab, as well as on our theragnostic concept. Theragnostic, of course, perhaps, Carmen, do you want to speak to that at the very, very, very high level, given where we are?
For the theragnostics?
Yeah. even as a general concept.
Yeah, so the idea of theranostics, I mean, I think you can imagine how this would work, but theranostics is a new, it's an evolving and a fairly exciting a branch of diagnostic imaging, essentially where people would take a scan. So if somebody had a scan, a lung scan or a scan for a neuroendocrine tumor or something, and in that those scans are usually set up such that the agent that's being used to scan, everybody's heard of a PET scan. So a PET scan works by essentially a sugar molecule is tied to a fluoride molecule injected into a vein, and cancers chew up sugar really quickly. So cancers tend to take up the sugar, and the sugar is made in a way that it's not really metabolized. So the cancers glow with the fluoride that's filled up. So that's a functional diagnostic scan. If at the same time you were to tie something to that sugar molecule and that fluoride molecule, like a chemotherapy or some kind of toxic agent, then you would be delivering treatment at the same time you diagnosed the location of the disease. So that's essentially theranostics. Now you can see all kinds of challenges with that. If I tie doxorubicin, if I tie a toxic chemical to my PET scan, then other things that take up that agent are going to get toxic treatment too. So our goal with our technology, of course, is to finesse that, is to be more exact and to be more precise. And you can imagine with a targetable hyperthermic therapy, we may be able to do that.
Yeah. Yeah. So exciting things coming in the future as well. Questions here about, financing. We raised money in the fall, and we've been very judicious with that cash and focused on getting human results with that. Post-human results, we will definitely need some more runway at that stage, but we think we'll be in a very strong position at that point. Associated with that, you have a question here on how exciting would this be within the industry in terms of if we have a really successful trial here and what it would mean for the cancer world. Carmen, you're best to answer that, I think.
Yeah, so this is, I'm going to just go right back to that first slide. There's 70% or 60%, if we're going to be very optimistic, of people on pembrolizumab won't respond. How excited is industry? If we could move that marker 10%, it would give them a new lease on life. If we can move it 20%, it'll be incredible. We are very confident we are going to have that impact at a minimum. And I think across all of those cancers you see, because this is not cancer specific. This is a biological process that we are applying to cancers across the board. And so the name of the cancer is irrelevant, which is the first, this would be like immunotherapy, the name of the cancer is irrelevant. The reason they don't all respond is because their biology is insufficient. They don't have proper antigen. We are solving a problem for that particular pathway, and we're creating our own new pathway. So yeah, they're excited. I presented, and we just saw the poster with Barry and I. We were in Vancouver presenting at the Canadian Melanoma Conference, and we were inundated. Not the first day, interestingly, people have to digest this. On the second day of the conference, we were inundated by interest and people recognizing there might be opportunity. And so David will tell you he's been fielding calls and managing we have to stay in step here, but there's lots of excitement about this.
Yeah, and sadly, the reality is there's a huge degree scope for improvement here. When you see a 34% survival rate, this particular study, lot of scope for improvement there. We should really have these two charts reversed order, right? Because it's the response rate that is first. Do you get a response? There's no guarantee that the 40% in this study that got a response actually survived 5 years. They just got a response.
Right. And that is back to that first question. You get a response doesn't mean you get a cure. And right. So, but increasing the response rate definitely increases the cure rate.
Yeah, and lots of scope in here, particularly with these cold cancers that we've talked about in the past, and what we think we can do for them. Tiny improvements makes a vast difference to because this is not representative of the population, of course, of people with breast and colorectal cancer. You juxtapose those numbers, and there's a tremendous need there for improvement, and that's what we're striving to do. So on that point, I think we will wrap it up. Thank you, everyone, for your time. We will keep you updated early on here, and in particularly when we get into this first in human study. There'll be lots of communications to make sure folks are apprised as to the status of it, and you'll hear our excitement, and we look forward to sharing that with you. Thank you, everyone. Appreciate it, and feel free to contact us anytime with further questions or around. Appreciate your time.
Have a great afternoon.
There we go.